Hello, everyone. My name is Joe Boyd. I'm a bioinformatician in the Fritz Lab at the University of Vermont, and I'm going to be talking to you today about my poster, Dimensional Reduction to Represent Multi-Layer Epigenomic Data. The subject of my talk is an R package that I've been developing. The R package is called ChIP-tsne. It's available at this URL if you would like to try it yourself. There's lots of different kinds of epigenetic sequencing data that's available now. We have ChIP-seq for transcription factors and histone modifications. There's also ATAC-seq, DNA-seq, and all of these techniques have something in common in that they rely on enrichment of sequencing reads to identify significant areas of interest. It can be really challenging to bring all these kinds of data together into one place to start reasoning about them, to start exploring them. And that's the motivation for the development of this R package, ChIP-tsne. I'm going to gloss over the details for the chemistry of ChIP-seq, but suffice it to say ChIP-seq and related techniques such as cut and run and ATAC-seq all result in enrichment of reads aligned to areas of interest on the genome, and we use statistical techniques to call peaks across the genome based on the enrichment of those reads. This kind of visualization is called a track visualization, and we're looking at different enrichment profiles for these histone modifications, transcription factors, and even RNA-seq and DNA-seq. All for this single location on the genome. And you can see that there are several different combinatorial patterns of these enrichment profiles just in this one region of the genome. And when we're analyzing this type of data, we need to consider tens of thousands of regions of the genome at the same time and all the different combinations and what their biological implications could be. So when you do one of these sequencing experiments, it's not just one location on the genome you have to think about. There's many thousands of them that will come back as being potentially of interest. And it can be really challenging to start reasoning about this data and getting a handle on it. We're trying to determine what are the biologically meaningful patterns, as well as what different types of enrichment profiles might be present at these locations. And particularly in more complicated experiments, this can be a real challenge. I was initially motivated to try to use tsne in this way by a talk that I saw by Lorenz van der Maten at a previous ISCB meeting. This was one of the images presented in that talk. And what this is 40 individuals' faces in 10 different photos of each, I believe. And the tsne technique was applied to the pixel values of all these images. And you get different patterns of individuals' faces clustering. And the technique does an excellent job of separating both individual faces while picking up on more subtle differences here, like the expression of this person. There's two different types of expressions that this person is making. So the inputs for ChIP-tsne are simply some kind of signal, which is typically from BAM pileups, but you can also supply your own bigwigs as well if you've processed the data in that way. And then you also need to supply regions to look at. This is typically peak calls or merged peak calls from multiple different samples, but it could also be something like a transcription start site or other relevant type of region on the genome. So you take your signal and your features, and we need to assemble a matrix for input into tsne. The way this is done is each feature will be given a row on the matrix, and we're going to stitch together bend values along the different samples going into our analysis. So in this pretend example, there are 5 bins for 3 different samples 5 times 3, that yields 15 different dimensions. And t-SNE is going to allow us to represent the patterns in this data in 2 dimensions. And again, the package does support UMAP if you're interested in using that. It's the same idea. So the data I'm going to be talking about is primarily H3K27 acetyl, which is a histone modification associated with active chromatin, cell line data in which transcription factor Icaros, XEF1 here, it has a heterozygous dominant mutation, and we're reintroducing wild type Icaros in an induction. So we're going to be comparing the induction of wild type Icaros to GFP control. This first view here is of this is a kind of replicate comparison, just a quality control type of simple view. And there's 2 replicates of H3K27. This is the GFP, and we can see immediately in the orange and purple, they agree with each other really well. There's kind of a lower enrichment domain over here and a higher enrichment domain of the t-SNE over here. And then within this higher signal domain, particularly, there's different shapes to these peaks. Some of them are double peaks. Some of them are more biased to one side or the other. So this could be potentially relevant to their orientation at a transcription start site or other kind of site on the genome. This other view, it's the same data, but here we've faceted the 2 replicates, and we're just visualizing the maximum pileup. Now, this is normalized data. In reality, one of these replicates, replicate 2, is much stronger than the other. And that's reflected if we visualize the peak call for these 2 replicates. There's a lot of peaks that are exclusive to replicate 2, although the vast majority are shared between the 2 replicates. And the peaks that are unique to replicate 2 are more biased toward this lower enrichment area of the t-SNE, which makes a lot of sense. If one of the replicates is weaker, you're not going to have enough statistical power to call peaks reliably there. In this application, we're doing these are differential results comparing the induction to the control. This first view here in A is looking at the agreement between control and induction. And B, we're looking at the difference, a subtraction from induction over control. So areas where induction is stronger are in purple, areas where control is stronger are in green. And then in C, we're doing the same kind of summary profile that we had shown previously, but overlaid with statistical differential calls from Seesaw, which is our package for doing differential comparisons in ChIP-seq. This is just a quick detailed view so you can better see the differences in this local region. This is the most complicated t-SNE projection that I'm going to get into. There's K27 acetyl, K4 monomethyl, the Icaros transcription factor, ATAC-seq, all 4 of them in 2 conditions visualized at the same time. And we're already zoomed into a small area of interest where it looks like something dynamic is going on with the Icaros, the ATAC-seq, and also K27 acetyl. To assign some kind of biological meaning, we can take the regions selected in this way and put them into something like the Great Enrichment Analysis tool. And when we do that, we get a nice list of relevant biological process ontologies. And we can even narrow down to a single locus here and look at what's going on more broadly in this one region. And this is the type of workflow that we envision will be really helpful when working with ChIP-t-SNE. Thank you very much for your attention. I'd be happy to answer any questions you might have through the Juno app. Thank you.